All right. So, welcome everyone online also. It's a great pleasure for me to introduce our speaker today in the QLS CAMBAM seminar series, Ivette Perfecto from University of Michigan. And Ivette is the James Crowfoot Collegiate Professor of Environmental Justice at University of Michigan. And I wish I had the word environmental justice in my title. That's just a fantastic title to have. So Ivette received both her master's and PhD from University of Michigan, and I don't have that in my note. Yvette, you were supervised by John Vandermeer, or you?
No, John Witter.
Okay, all right.
Yeah, but I do
'Cause
collaborate with
you
John
collaborate a lot
lot.
with John Vandermeer. Yes. Yes, so she obtained her PhD in University of Michigan in ecology and natural resources. And she was, still recently elected member of the American Academy of Arts and Science in 2023. And her lab, so Yvette has done just is a complete ecologist to me when I look at her work, from doing a lot of developing theory in ecology, really fundamental research, to working on agroecosystems and doing outreach, research, education, covering all the angles of what an ecologist can achieve in our field, in our discipline. So our research is really interesting, studying agroecological research in the neotropics and in North America, focusing on biodiversity in agricultural landscapes. That's the, I think, current focus of a research. Works on spatial ecology and coffee, agroecosystems, and urban agriculture. And I think so today she will talk to us about ants a lot, focusing on ants.
We lost the PowerPoint.
We lost the PowerPoint. No. Oh, yes, we lost the PowerPoint.
Hi, everybody. Sorry for the technical difficulties, but
We're good.
Over to you, Yvette.
We're good. Okay, I hope it doesn't go back in the middle of the talk. Hi, everybody. It's a pleasure for me to be here. Thanks for the invitation. So today I'm going to talk about ants and ecological interactions in agroecosystems. And just to give you a little bit of context, I consider myself an agroecologist. I want to thank, before actually starting my talk, I want to thank a lot of people that are responsible for this research. It's not my exclusive research, of course. There are many graduate students in my lab. John Vandermeer, as was mentioned before, is my colleague, and we work together, and some of his students have participated as well. And I want to also recognize the coordinators of our project in Puerto Rico and Mexico, and of course the farmers that allow us to work on their farms. As I was starting to say, I consider myself an agroecologist, and agroecology is considered to be a practice, a movement, and a science. And today I want to talk about the science of agroecology. So the science of agroecology, I define it as the study of interactions among organisms, their environment, and humans in agroecosystems, with the purpose of designing and managing them in a sustainable way. Within that broad scope, I am interested in ecological interactions that might contribute to reducing pests and diseases in the agroecosystem. One of those groups of organisms are ants. I have done work with ants for many, many years, and ants can have a very complicated relationship with pests and diseases of crops. Their effects can be positive, but they also can be negative or neutral. But for the most part, they tend to be positive. So it's good to have lots of ants in your farms. Now, our work is rooted in the dynamic interplay between natural history and theory, two elements that are intimately interconnected in our philosophy of research. Natural history, that is usually acquired through field and laboratory observations, serve as a foundation or the foundational context that informs our models, while the models in turn offer insights into the complexities of nature. And we believe that both components are equally vital to our understanding of nature. Our commitment is to present a unified narrative that honors both the natural world intricacies and the theoretical frameworks that we develop to interpret them. So I say this to give you a context of where I'm coming from and the importance of the natural history component of our work, as well as the theoretical component. Okay, so as I mentioned before, I consider myself an agroecologist, and as an agroecologist, I work in an agricultural systems or in agricultural systems in general. For many years, I have been studying the ecology of the coffee agroforestry system. What you see here in this image is a coffee agroecosystem. Coffee is a great model system because it can be produced in a variety of systems that range from very forest-like systems like this here, you see in this extreme, what is called sometimes rustic coffee, all the way to coffee monocultures. So it's kind of a gradient. You find these gradients frequently in landscapes, coffee landscapes. Now, a few years ago, we started a project trying to understand the ecology of the ant community in coffee farms in Puerto Rico, as well as their ecosystem services of pest control in particular. And that's in part what I'm going to talk with you about today. Now, we frequently sample ants using tuna baits because we're interested in the ants that are kind of predatory ants that will go for tuna baits. So we sample tuna using tuna baits. And in this particular study, we sample the ant community in about 25 farms throughout the coffee growing region in Puerto Rico. And here are listed the species that we encounter. Constructing a species rank graph that you see here to the left, you can see that there are 4 species that are disproportionately represented, have a high representation. However, only 3 of them were dominant in at least, so this here, these are the four species that are overrepresented, but 3 of them are the ones that are dominant in at least half of the farms that we sample. And these 3 species are the following: Wasmannia auropunctata, which is also called the little fire ant or the electric ant; Solenosis invicta, which is the red imported fire ant; and Monomorium floricula, who sometimes is called the flower ant. Okay, so we began by looking more in depth to the interactions between these ant species, so the competition between the ant species. Now, competition has been described as the hallmark of an ecology. And this review by Cerda confirmed the importance of competition in structuring ant communities. Furthermore, they tend to form these competitive hierarchies. So there's a lot of documentation in the literature about the competitive hierarchies that function in a transitive fashion. Now, you're all quantitative biologists, so you're probably very familiar with transitivity, but just in case you aren't, for those who are not familiar with the term transitivity, the prime example of transitivity or a transitive characteristic is height in humans, no? If Gfan is taller than Cara and Cara is taller than Rosa, then Gfan is taller than Rosa. So that's a transitive characteristic. In competition, this will be, for example, if species A beats species B and species B beats species C, then species A will beat species C. No, that's the transitivity in competition, and usually they form a hierarchical structure where eventually one species is the dominant species. However, competition does not have to be transitive. It can be intransitive, like the game rock, paper, scissors, no? You have the illustration here of the rock. I'm sure all of you are familiar with that. Based on field observations and some natural history, we suspected that the three most abundant species, the ones that I just mentioned to you in the coffee farms in Puerto Rico, engage in an intransitive competition, where Wasmannia here beats Monomorium, Monomorium beats Solenopsis, and Solenopsis beats Wasmannia. So let's examine then the evidence for this. And to do this, we usually look at pairwise interactions. There's a lot of work about intransitive competition in the plant literature, and it has been documented. There's a fairly large percentage of interactions that are intransitive. Now, in terms of the ants, we will look at the pairwise interactions between the species to figure out, whether we have transitivity or not, or whether we have an intransitive loop, as I am proposing here. So let's first look at the first interaction, which is Wasmannia and Monomorium, and basically on studies that have been published. These are data from a pairwise nest box competition experiment between Wasmannia auropunctata and another species of Monomorium, not Monomorium floricola. And these were conducted by Volchak and colleagues, these studies. In this study, although Monomorium recruited faster and retrieved more resources than Wasmannia at the beginning, at the end of the experiment, Wasmannia ended up killing and eating the Monomorium. And so here you have the average number of dead workers, always lower. You have lower for Wasmannia auropunctata as compared to the Monomorium, but most importantly, at the end, these are another set of experiments that they run for a longer period of time. And as you can see, this is the proportion of live workers after 12 days. And you can see that in basically of the 9 trials, in 6 of them, Monomorium workers were completely exterminated. Basically, the Wasmannias ate all the brood and the workers, and including the queen as well. So very strong competitive effect of Wasmannia against Monomorium. Well, in this case, it's actually predation. So now let's look at the interaction between Monomorium and Solenopsis. Now, this is a little bit a harder, saying that Monomorium beats in competition Solenopsis is a harder pill to swallow, because for those of you who know ants and know these two species, Monomorium tend to be a very docile species, not extremely aggressive, at least to the human eyes, and Solenopsis invicta is a very aggressive ant. It recruits a lot very fast and has very large nest. And so you will think that Solenopsis will beat Monomorium in competition, but the reverse is the case. And it turns out that Monomorium floricola, as many other Monomorium species, have a very important weapon that they use very effectively against Solenopsis. And here I want you to watch here this little ant. This is the Monomorium, and these are the Solenopsis here. But I want you to look at this Solenopsis invicta and this Monomorium interacting. So you look at the abdomen, the gaster, and she's spraying something on the bigger ant, on the Solenopsis. And at the beginning, it doesn't look like it's being very effective, but eventually the Solenopsis start trying to clean its antenna, but eventually it just run away. And then I want you to look at this one. I'm going to play this again. I want you to look, oops, sorry. I want you to look at, let's see here, at this one and the abdomen. No, that I showed you before. I want you to look at another ant, this one that's coming here, and another Solenopsis who will be coming from up here, that one, and look at the abdomen and its motion, you know, how it turned its gaster toward the ant, and it goes like that, and it spray the other ant, the Solenopsis, with this venom, and the Solenopsis immediately go away. So it turns out that the genus Monomorium produce at least these two venoms. These are alkaloids to protect themselves against other ant species. And we have some data also from baitings that were conducted in citrus trees that were intercrop with coffee. This is still in the coffee farms. And as you can see, these are just examples of many, many baitings that we conducted, but these are typical examples where sometimes both of them start increasing, but eventually the Monomorium, which is in blue, will win in competition for that resource against the Solenopsis, no? So this is the most common thing that happens. Every once in a while, when the baits are very low in the trunk of the tree, not up in the branches, but in the trunk, and closer to the ground, you have an outcome that looks like this, where the Solenopsis invicta start recruiting really, really fast because they nest on the ground. And so they can start recruiting really fast, and they actually overwhelm the Monomoriums. The Monomoriums are never able to get a good hold at the resource when the resource is placed really close to the ground. But on the branches, basically, this is the main outcome. So that's the relationship between Solenopsis and Monomorium. And now we have to look at the interaction between Solenopsis versus Wasmannia. And again, here, what we are expecting in this kind of interaction, for it to be an intransitive loop, as I am arguing, Solenopsis has to win in competition against Wasmannia. And that is what this is showing here. These are results from a team from China that has been looking at interactions between these species of ants. And they have a lot of evidence that that Wasmannia wins, basically, no, sorry, that Solenopsis wins in competition against Wasmannia. And you can see here, this is the percent, the mean percent mortality of the ants, having when you have the ants one-on-one, meaning not one individual to one individual, but when you have a proportion of 1-to-1. So if you have 50 Solenopsis, you have 50 Wasmannias, no? So a 1-to-1 relationship, you have lower mortality in Solenopsis than in Wasmannia. When you have a 1-to-2, so that's for every one Solenopsis, there are 2 Wasmannias, you still have, essentially, you still have Solenopsis winning competition. When it is until you get a proportion of 3 Wasmannia to each Solenopsis that you then get a neutral competition. Now, there's no winners in this one, no? And so that's some of the evidence, and we have conducted some of those nest box experiments. Here you can see the setup where we have Solenopsis in one, Wasmannia in another one. We connect them with these bridges, and then we wait for a week or so to see who wins in competition. And basically, in these nest box trials that we have conducted, Solenopsis displays Wasmannia in 9 out of the 9 trials that we have conducted. So basically, we have some evidence, some experimental evidence that this could be an intransitive competition between these species, or among these species. Now let's move on to our field site. in the coffee agroforestry system, there are usually 3 habitats for the ants. Basically, you have the ground or leaf litter, then you have the coffee bushes themselves, or any other bushes that are, not very tall, and then you have the canopy trees and the higher canopy of trees. In this particular case, in this farm, we have a lot of orange trees, and so some of them, they don't grow really, really tall, but we also have some shade trees that have much higher canopy. So anyway, what we found is that Wasmannia is found all over the place. Basically, you can find Wasmannia foraging on the ground, you can find Wasmannia on the coffee and the layer of the coffee bushes, and you can find Wasmannia up in the canopy of the trees as well. Solenopsis will forage on the ground, they nest on the ground, and they forage on the canopy of the trees as well as in the ground. I forgot to say that Wasmannia can nest anywhere in the leaf litter, in the coffee bushes, or up in the canopy of the trees as well. And then finally, Monomorium is an arboreal ant. So they nest within the coffee, within either the coffee bushes or the shade trees or the orange trees, and they forage in the canopy as well as in the lower parts of the bushes, or within the coffee bushes. Okay, so this basically means that there is a potential of interaction between these 3 species at the layer of the coffee bushes, which is where we have been conducting our sampling, no, at the layer of the coffee. And so that's where you can have this intransitive loop basically functioning. So given the pairwise interactions and the fact that these 3 species occupy the coffee bushes, can we see any evidence of intransitive competition in the field? And to examine that, we set up a plot in Finca Gran Batey, which is one of the farms where we work in Puerto Rico, and sampled the ants foraging on each of the coffee bushes. The samples are taken by putting tuna fish, 5 baits of tuna fish on the coffee bushes, and then going back half an hour later and identifying the ant that is occupying that bait. So this plot here is approximately 30 meters by 120 meters. And here we can see the distribution of the 3 species. Wasmannia is in green, Solenopsis is in red, and Monomorium is in blue. And the size of the bubble represents the proportion of the baits that are occupied on each plant. And as you can see here, the plot is dominated by Wasmannia, with some scattered bushes dominated by Solenopsis and a patch that seem to be dominated by Monomorium around this corner here of the plot. But this is a snapshot, and a snapshot does not tell us much about the dynamic, no? It doesn't tell us anything about the dynamics. And so we need to see how this changes over time. If the intransitive competition is indeed happening, we would expect areas of Wasmannia to be taken over by Solenopsis, and areas of Solenopsis to be replaced by Monomorium, and areas of Monomorium to be replaced by Wasmannia with time, no? And so we decided to continue sampling this plot. We started in January 2019. And here you can see the sequence from January 2019 to January 2024. So 5 years with an intermission during the pandemic that we couldn't sample. So we sample in the dry season and the wet season. So in January, February, or December, January, and then again in June and July. And what you can see here is a clear replacement of Wasmannia by Solenopsis. In these areas that were dominated by Wasmannia eventually ended up being dominated by Solenopsis. A weak, kind of subtle takeover of Solenopsis by Monomorium in some areas, like here, as you can see, and here to some extent, and a more delayed takeover of Monomorium by Wasmannia here. So Wasmannia started entering through this corner of the plot and eventually replacing or eliminating the monomorium. And so this seemed to be playing out with all, the variability that you find in nature, but it seemed to be playing out very nicely in the field. And so that was for up to now, I've been talking about the natural history that inspired the theoretical models, no? So we have some evidence that this intransitivity between these species is real, no, it's happening. And a lot of what I'm going to talk about in the next few minutes come from these papers. Two of them you have read ahead of time. And one of them is this one here is a paper that John Vandermeer published long time ago in 2011 on ecological complexity. And then the other two are more recent papers that based on this particular system. And as I mentioned before, we have this triad of intransitive competition, which forms this intransitive loop. We can model that system with a very simple Lotka-Volterra equations with a single parameter, which is alpha, the competition coefficient, and the competitive effect in this particular case, we're assuming that is symmetric. So all the alphas are the same. Again, very simple model. These generate, if you model this set of equations, it basically generate what is called a heteroclinic cycle. That means that the system moves from dominance of one species, dominance of the other species, to dominance of the other species, and it continues going in that direction, getting closer and closer and closer, to complete dominance of one of those species. Eventually, with an heteroclinic cycle, eventually one species will dominate, but it can take a very, very long time for that to happen. This is when the competition coefficient is high. When the competition coefficient is low, then you end up with a focal point, like in this image here. So you either have a heteroclinic cycle or a focal point, depending on the alphas. If the alphas are low, then you can get a focal point, and if they're high, then you get a heteroclinic cycle. And this is the modeling here. This comes from John's paper from 2011. And essentially, this is what this is showing. And here are the outcome of the model, where you have the species basically surviving, all the species, the three species together, but moving farther away with higher and higher fluctuations. If this is it, when the alpha is more than higher than 2. When the alpha is 2, you have these neutral cycles, and then when the alpha is higher lower than 2, then you have the focal point. Now, going back to our system, in Puerto Rico, the three species in the intransitive loop are not the only species present. Here, we show a list of other species that were recorded in our sampling of the coffee plants. And there are 13 additional species that have been recorded, some only 1 or 2 times, others more frequently, no? And so we started questioning, how are these species then maintained in the system, no? And in our paper in PNAS, we propose that another mechanism for maintaining species diversity is this intransitivity. Remember how I mentioned that dominant hierarchies were common in the ant communities. When you have a dominant hierarchy, then you have a species that eventually can basically extinguish all the other species because it's winning in competition. So we ask, what happens when you attach a dominance hierarchy to an intransitive loop? So you have this intransitive loop of the 3 species, no? And so what would happen if you attach to one of these species and a dominance hierarchy? Like, for example, here we have a hierarchy of species 4 beating species 5 beating species 6, and the Wasmania, which would be species 3 here, beating species 4. And it's a dominance hierarchy because Wasmania will beat all of the other ones. Species 4 will beat species 5 and 6, and then species 5 will beat species 6, no? And so that's the dominance hierarchy. So what happens when you attach that dominance hierarchy to this intransitive loop? And here are the equations. Again, these are the three equations that I showed you before. Then we added the one for the 4th species, and then connecting them together in a hierarchy, in a competitive hierarchy. And so when you model something like this, essentially, well, this is the transitive structure, and this is the intransitive structure of the model. And so to the left here, A, B, and C is what I showed you before. What happens essentially is basically the same thing. When you add a competitive hierarchy to one of the species in the intransitive loop, essentially, if the if the alpha is higher than 2, if you have strong competition, then you end up with an heteroclinic cycle with 6 species, with the 6 species basically being part of the heteroclinic cycle. If the alphas are low, so if you have an alpha lower than 2, then you basically have a focal point. No, you have the species basically get to an equilibrium point, and you maintain all 6 species in the system. That's the point, no? That the intransitive loop, the intransitive competition, basically facilitate the maintenance of the species that are in a hierarchical structure. If it wasn't because of this intransitivity here, essentially what you would have is that let's say that you didn't have the Solenopsis, or let me go back here. Let's say that you didn't have the Solenopsis or the monomer, you only had the Wasmania, and you have Wasmania in this transitive hierarchy. Eventually, Wasmania is going to dominate the system, and you're going to end up with a system of one species, no? Because Wasmania wins in competition over all the other ones. However, because Wasmania is part of this intransitive loop, when Wasmania is winning in competition, then the Solenopsis comes and beat it down, no? And so it releases the other ones from competition. And then eventually it happens again. So the intransitive loop helps maintain the 6 species in the system. It's a mechanism for maintaining species diversity. So you can imagine, this is the intransitive loop, and you can imagine each one of those species actually having a transitive hierarchy attached to it, and that could be a mechanism of maintaining all those species diversity, or they can even be connected, or you can have an intransitive loop of 5 species, and some of those species have their transitive hierarchies, and those can be connected as well, etc., etc. So you can imagine all these interactions, and that or all these species that can be maintained in the system by the intransitivity. Now, let's go back to the natural history. As I mentioned before, my work basically is a back and forth between the theory and the natural history. And okay, so we look at transitive tails attached to an intransitive loop as a potential mechanism for coexistence, no? But natural systems are always more complicated than what we imagine them to be. And in our system, it turns out that one of the intransitive species, that is Solenopsis invicta, has a parasitoid that changes its behavior. That inspires us to ask what or how do higher order interactions, like this one, affect intransitive competition. and also what are the implications for species coexistence of these higher order interactions. And the reason I call this a higher order interaction is because it basically the parasitoid had an effect on the behavior of the ants. Here I'm showing you a piece of we shifted from tuna fish to hot dogs. And so this is a piece of hot dog that is about half a meter from the nest mound of Solenopsis invicta. And you can see that they're recruiting a lot, they're very much active there. Now, here is looking at the same nest, exactly the same nest, exactly the same distance, but in another direction, the same day also. And what you notice here is the activity is not that high, but you also notice these little flies that are all over the place. These are the phorid parasitoids, Pseudactyon. There are a couple of species. And so they have an effect on the behavior of the ants. The ants stop recruiting that much. They don't move as fast. They do that to avoid being detected by the phorid parasitoids and being parasitized. And so that is what we call a trait-mediated indirect interaction or indirect effect. And that is a higher order effect. And so what we decided to do then is to verify this with some experiments in the laboratory. We basically have these nest boxes with Wasmannia and Solenopsis. We connect them together, and then we look at the outcome. And here, what you can see is that these are the number of Solenopsis that were found in their own nest box after a couple of days of interaction. And you can see that if there were phorid presence, if the phorids were present, you have a higher number that were found on their nest box. So they basically stay where they were. But when the phorids were absent, basically they left their nest box. They moved to the other nest box, the Wasmannia nest box. And here you can see the number of Solenopsis found in the Wasmannia nest box. So you have a lower number when the phorids are present, and when the phorids are absent, no, you have basically a higher number. So when the phorids are absent, when the phorids are not there, they behave in the normal fashion very aggressively toward Wasmannia auropunctata. When the phorids are there, they just stay put on their nest and they don't forage that much. And here you can see this is the number of Wasmannia. Basically, the phorids had no effect on the Wasmannias. But one thing that I want you to notice here is the number of Wasmannia that were found in their own nest, because here inside these nest boxes, you have basically the ants. I put a little structure for the ants to be able to put their brood inside. And basically what happened is that when the phorids were absent and the Solenopsis were very aggressively moving toward the nest box where the Wasmannias were, the Solenopsis took over the nesting site of the Wasmannias and basically kicked them out or started fighting with them, and the Wasmannias were still on their nest box, but on the edges and trying to hide their brood from the Solenopsis. So basically, we have evidence for that happening. And so let's look then at the model. So here as is a representation of the model, it kind of a graphical representation where you have the intransitive loop. This is the same as in the previous model. So you have Solenopsis beating Wasmannia, Wasmannia beating Monomorium, Monomorium beating Solenopsis, and these are the alphas. But then attached to the Solenopsis, we have a predator-prey interaction with the phorid. So you have the regular density-mediated predator-prey interaction, but in addition to that, you have a trait-mediated effect. So the presence of the phorid changes the behavior of the ants, affecting the interaction between the Solenopsis and the Wasmannia. And so we model that with this set of equations. And basically what you have here, we added this term, which is the functional response term for the predator-prey system. And so this is the trait, the density mediated effect. So with a functional response. And then we also have a trait mediated indirect effect that is represented here, no? So basically we added that to the set of the three equations for the intransitive loop. The outcome, basically, as I mentioned before, for the intransitivity, this is the same. You know, we have this a focal point outcome. This is just for the three species, the intransitivity. You have the focal point for the low competition when alphas are low, and we have the heteroclinic cycle when the alphas are high. So that's the same there. But to that, now we're adding these predator-prey interaction, predator-prey oscillations. So essentially what we're talking about here is a system of two coupled oscillators. One oscillator is the Solenopsis phorid system, predator-prey system, and the other oscillator is the intransitive loop that goes from monomorium to Wasmannia to Solenopsis to monomorium to Wasmannia to Solenopsis, et cetera, no? So that's another type of oscillator. And these are connected, no? Through the Solenopsis. And so a triad oscillator and the predator-prey oscillator. And I'm sure that you're familiar with the two oscillators that are connected. So physically, you can kind of use the metaphor of the pendula, which is used very frequently in ecology to basically talk about these oscillators. So you have the predator-prey oscillator, and then you have the oscillation between Wasmannia and monomorium connected through the Solenopsis, no? And so here you see, you know, well, if the connection is a loose connection, if it's a rigid connection, you just have that oscillation. But if it's a loose connection, like what we have, then this is what you get. And what that means is basically chaos. You have a chaotic behavior, chaotic outcome when you link these two oscillatory systems. And so this is a system of two connected oscillators, and this is the outcome of the simulation. You can see, well, this is basically a chaotic system. And but even though it's a chaotic system, it has some regularity to it, no? Chaos doesn't mean necessarily that it's completely irregular. There are some patterns in this particular outcome. You can see some patterns, but formally speaking, is chaotic, is a chaotic system. So you're never gonna be you're never gonna be in the same trajectory. And here I'm showing you the trajectories between monomorium and Wasmannia, Solenopsis and monomorium, Solenopsis and Wasmannia. And here are the four species, including the phorid, no? The phorid is this one here in the orange, and the Solenopsis is the one in the blue. So essentially what we have is really cool, I think, because what you have is a chaotic system that, you know, people tend to think, oh, it's chaotic, so it's unpredictable. Yeah, you cannot predict exactly where it's going to be in the future, but there is a pattern to this chaos, no? And so that's what is cool about it, is that the chaos has a pattern. And what I did here is just expand it a little bit so that you can see a little bit better the oscillations. And what what you have is essentially these higher oscillations between the phorid and the solenopsis, no? These higher oscillations here, and or let's say a little bit longer term oscillations between the phorids and the solenopsis, but then in between those oscillations, inserted within those oscillations, are the triad oscillations of the intransitive loop. And so, and here I'm showing you, let's see if it, yeah. So this is just one cycle, no, just expanding this from this peak to this peak, let's say, no? And you can see that, you know, you have this longer term oscillation of the phorid with the solenopsis, but in between that, inserted within that oscillation, is the oscillation of the triad of the intransitive loop. And so to summarize, empirical demonstration of intransitive competition among the three dominant species in the coffee system in Puerto Rico, I hope I have shown you that there is at least some empirical evidence for that, both in terms of pairwise experiments and pairwise comparisons between the species, but also in the spatial pattern that they form in the field. And then we, using that as an inspiration, we then developed these models of intransitivity as a mechanism for the coexistence of species. So what you have here is that a way that that can happen is by combining an intransitive loop with a transitive hierarchy, as we did, as I showed you before, but also adding a predator-prey oscillation with a trait-mediated effect or a higher order effect that also maintain all the species in a chaotic system, basically in a chaotic outcome of it. So some of you that are more inclined to be very practical might say, well, so what? Okay, yeah, we understand better. And, you know, I think that that's good enough. But I'm also interested in the practical implications of this. And as I mentioned to you before, these ants are important predators of the main pests in the coffee system. So here you see the coffee berry borer, which is the main insect pest in coffee all over the world. And then you have this blotches here are made by the coffee leaf miner, which is another very important pest in coffee farms. And so the ants are very important predators. The bigger ants will eat this beetle before it gets inside the nest, inside the berry. It takes this berry borer about an hour to get all the way through and be protected from the bigger ants that cannot go inside the hole. But you know, you have in the system, you have ants that are of different size, and I'll mention that to you because these ants that we have in this system in the triad of the intransitive competition have different sizes. So here you have, for example, the Wasmannia repunctata is a really, really tiny ant, and it's almost the size of the berry borer, and they can penetrate the holes that are made by the berry borer and eat the brood inside the berry borers. The Solenopsis invicta, or Solenopsis invicta is a much bigger ant and cannot penetrate, but they can easily grab these beetles and take them away before they get inside. And so you have a different, you know, because of this difference in size, Monomorium is also a little bit bigger than a little bit longer than Wasmannia, but it can also penetrate. So you have, you know, different effects of these ants on the pest. Some of them, the bigger ones, can take the berries out, you know, before they get inside, so they can prevent damage, no? And the small ones can get inside the berries and eat the brood and affect them also by eating the brood. Now, one of the problems with having the Solenopsis is that while they can protect the berries from getting damaged, those berries that they miss for one reason or another, but the plant is still basically dominated by Solenopsis, if the plant is still dominated by Solenopsis, it basically keeps out the Wasmannias and the monomoriums. So essentially it protects, if the berry is able to get inside, if the berry borer is able to get inside the berry, then it's protected because the smaller ants cannot go to these plants, to these berries, because the bigger ants, the Solenopsis, is monitoring those plants. And so there is a kind of a dilemma there in terms of the impact of these ants. So when you have the larger ants, the Solenopsis invicta, coffee bushes that are dominated by Solenopsis invicta are expected to have lower damage, but higher survival inside the berries, no? So the coffee berry borers will have a higher survival and lower amount of damage. And if the plant is dominated by either Wasmannia or Monomorium, we will expect lower survival inside the berries, but perhaps higher damage, because they're not as effective in being able to grab the insects, no, themselves. And so these are experiments that were conducted by one of our students, Janice Newson, and you can see that the plants that were dominated by Solenopsis, this is if Solenopsis was present, basically the plants, when Solenopsis were present, the plants have lower damage, but they have higher survivability, although in this particular case, it was not a significant effect. And then if they were dominated by Wasmannia ropunctata, basically Wasmannia ropunctata have, the ones that were dominated by Wasmannia ropunctata have a higher survival of the brood inside. No, with the brown that were present have lower survival of the brood inside the berries, no? And so this actually plays out in the farm. Patches that are dominated by one species versus the other are going to exhibit a different kind of damage, or more or less pest damage, depending on the pest that we're talking about. And so I'm going to finish just by saying the story of Don Bernardo. Don Bernardo was the owner of Café Gran Batey, one of the farms where we work. And this is where we have this plot, no? And when we first started working here, he say, oh, the ants are driving me crazy. The Wasmannia, which in Puerto Rico are called abayarde, are coming back and they're all over the place. This is the paradise for the abayarde, paraíso de los abayarde, and this is a problem because according to him, and I have experienced this as well, these ants sting, and they sting really badly. And they're so tiny that you don't notice them, but then once they get into you, they bite you in the soft tissue. And so it's really hard for the workers that are picking up the coffee to be in these patches where there are a lot of abayarde, a lot of the Wasmannia ants. And so he decided that he was going to spray something for the ants, and he sprayed diazinon, which is a very nasty chemical, and he was very happy. This was before we started working there, actually. He was very happy because the abayardes were gone, but so were other ant species as well, including the Solenopsis. And essentially what happened is that after he sprayed this, the next year he got an incredible outbreak of the coffee leaf miner. Essentially, by eliminating the ants, he basically got rid of some of the most important predators of the coffee leaf miner, and he had an outbreak of coffee leaf miner that affected the production. And so again, it's important to maintain the ants in the system, and our study is actually giving us some insights as to why you find this patchiness in terms of the pests themselves. No, you have sometimes areas where you have a very low damage of the coffee berry borer, but then in the same farm, within the same farm, you have a patch where the coffee berry borer or the coffee leaf miner is very abundant. And so understanding the relationship between the ants and the pests, and also the spatial distribution of the ants, and what is the dynamic of these species in terms of their spatial distribution, can give us some better understanding of this interaction with the pest. And perhaps we haven't gotten to that stage yet, but perhaps will help us design a system that might encourage the species that are more favorable for controlling the pest. And with that, I want to thank you and open it up for questions.
Thank you, Ivette. We have questions. From the audience, yeah, try.
Can you hear me?
Can you hear, well, talk and we'll see if,
Can you hear me?
okay, so yeah. Move closer. Sorry, I have to move closer to
Can you turn up the volume on the microphone?
the,
Yeah. And if you please ask next to the microphone and let me turn it on.
Where's the microphone?
Right there.
Okay, yeah. So I have two questions. The first one is on the page of the two coupled oscillators. Could you please share that?
Uh-huh. Okay. Are you still seeing the slide?
No.
No.
Okay, so I'm gonna have then to share again. Okay. Can you see it now?
Yeah.
yes.
Okay, a couple oscillators. Here?
Yeah, it's okay. So as the next one, the next, yeah. So I'm wondering why the second oscillator changes the first oscillator's direction. Does it change or and how?
Yeah, okay, so here you have the oscillation between the predator prey, so that means that the solenosis is the solenosis is going up and down,
Mhm.
basically, high solenosis and low solenosis. And it doesn't change the direction. I actually put this there just to show how, it can come up and down. But it depends on the competition coefficients that you include, no?
Okay,
So yeah, but it does change the number of the solenopsis. And so it depresses the solenopsis when the forts, when the forts, basically when it's in this area of the oscillation, and increases the numbers. Yeah, the density,
okay.
basically. But it doesn't, yeah, no, you're right. I shouldn't have put this here, because it depends on what are the competition coefficients.
Yeah, so like, is it possible if like it might change the direction or not, or never?
What is that again?
Like for the direction of the first oscillator, is it possible to change when you add the second oscillator? Change or not, or never?
I think it's possible, but only if you change also the competition coefficients. But if you leave the same competition coefficients, I don't think so. But I don't know, I have to simulate it to make sure.
Yeah, okay, thank you.
But theoretically, I'm not sure. I need to do the simulations to be able
Yeah,
to
that's
see
very
if,
interesting.
yeah, to see if it changed the direction. Yeah.
Yeah,
I mean,
thank you.
from a heteroclinic cycle to a focal point. Yeah.
Yeah, exactly. Thank you. And then my second question about the first relationship you give as an example of the two ants.
The what?
The two types of the ants, you give an example for the relationship between them. You give a molecular substance as a
Yeah, that's
E.
the for
It's my
the monomorium.
yeah. So because my previous study is also about the root paper sensor relationship, but it's in microbiology. So I'm wondering, because in microbiology, I think when I found some reason, some mechanism for the relationship, I can only go to the molecular way, but I think the metabolism network is very complex. So do you think the key mechanism for the relationship can be only one substance, or it could be a very complex work? Do you have any suggestion for the microbiology side?
I don't know, but I would imagine that it could be a number of substances. These are only two that have been reported, but
Mhm.
you know, ants, different species produce all sorts of. People are looking into these chemicals that the ants are producing, and there's a lot of work that has been done actually, and there are all sorts of other complex molecules that are produced as well, and potentially interactions between them as well. But yeah, but I don't know that literature very well.
Okay. It's a very fantastic presentation. Thank you.
Thank you.
See, comments in the chat. Oh, just some thank yous and great talks. Maybe I have one, so you have gonna have a longer discussion with the students. So I'll take the opportunity to ask myself a question. It looks from the on the applied side. So you took the predator-prey dynamics with the parasitoid. Is there an interest or to include the predator-prey dynamics of the ant with the pests, with the berry borer?
Oh, yeah. We haven't done that, but that's an excellent question that we're hoping to have a student. We have an undergrad student that's working with us now that is very interested in this system, and she's interested in looking at that particular, you know, interaction. And so, yeah, we haven't modeled that at all yet, but it's possible to do so. And you know, I think it would be very interesting as well to
The
examine.
rich behavior that's possible in there. Yeah.
Yeah,
Well-
no, definitely, yeah.
Well, see, other questions. I will thank you again and join me in thanking Ivette again for a great talk.
Okay, let me get out of here.
And I think that we are giving you a little break before discussion, and if